the basics of infection. So this is a little bit more specific to microbial infection. So start off with a couple definitions. So pathogenicity is the ability of a microbe to cause a disease. So if a microbe is called a pathogen, basically it means it causes a disease. So not every microbe is a pathogen. Like a regular strain of E. coli is not a pathogen because it doesn't cause an infection. It's a microbe, not a pathogen. Whereas Staph aureus can be a pathogen. Virulence is the extent of pathogenicity. So if something is said to have high virulence, that means it's very pathogenic. It causes infection very easily. So the three main portals of entry for microbial infection are the mucous membranes. Again, mouth, nose, eyes, so forth and so on. You can actually get infections through the skin, through either cuts in the skin or actually through the skin itself. And then the parenteral route, which basically means intravenously. So here's some of the terms that are used to describe how virulent a bacteria or virus is the ID50 and the LD50. Okay? The ID50 is how much of a microbe or how much of a microbial toxin does it take for 50% of the population to be infected. Okay? So it's usually a concentration of a toxin or a number of cells. LD50 is the same, but instead of just monitoring infection, it monitors death from a particular type of infection. So how much of a bacterial infection does it take for 50% of the population to die from that infection? Okay. So here's ID50 of Bacillus anthracus. So you guys can probably figure out what disease we're talking about here, anthrax. So again, anthrax is caused by the endospores of this bacteria. So that's the infectious part. It's not the bacteria itself, it's the endospore from the bacteria. So you can see based on the portal of entry, so through your skin is the easiest way to get infected from these spores. So the ID50, again, 50% of the population would get an infection between 10 and 50 endospores, so very few endospores. Inhalation takes a little bit more, it takes 10 to 20,000 endospores for 50% of the population to be infected. Ingestion, so basically ingesting it either by drinking or eating it, can take up to 1 million endospores. So that can be, again, a number of bacteria, a number of spores. Another way you can look at it is toxins. So some of the disease-causing agents that have to do with microbes aren't the cell itself, but it's the toxin that the cell makes. So here's some toxins that are made from bacteria. Botulinum, the Shiga toxin, again, that's from Shigella, but also from the E. coli strain that picked up that gene, and a staph toxin. And this is basically the concentration required to cause infection in 50% of the population. I'm just curious, does anybody know what botulinum is used for?
Botox?
Yeah. So that's the toxin that's what Botox stands for, botulinum toxin. So now we'll kind of go through how physically does the infection happen. So now we've kind of gone through how the bacteria or virus can get into the body. Now we'll talk about, okay, well, what happens after it gets into the body? So the first thing it does is it usually will infect cells or attach to cells in the host. Okay, so that's called adherence. So there's usually some proteins on the outside of the bacteria called adhesins or ligands that bind to receptors on the host cell and basically cause the bacteria to anchor down onto the cells of the host. And then they start to form biofilms on various, in the organs and different places in the body. So here's a picture of how that looks like. So here's a host cell. Okay, let's just say this is a liver cell. And then here's a bacteria that has these little adhesins, these proteins on the outside, and they bind to these receptors on the liver cell and basically start to anchor down on the liver and then start to form a biofilm, start to grow as a film on top of the liver. So here's just a couple examples. E. coli on urinary bladder cells. So you can see they're kind of sticking to these cells. Here's some bacteria adhering on the skin. This looks like staph. So now, basically, the bacteria is anchored down in the cell, and now it tries to penetrate the host defenses. And basically what that means is how does it prevent the immune system from eradicating it immediately? And there's a few different techniques that bacteria use to do this. So some bacteria have capsules, which is like an outer covering outside of the cell wall. We've talked about like the glycocalyx as a capsule. So it's like a protective covering. And what that does is prevent phagocytosis. Phagocytosis is basically we have cells of the immune system, and we'll be talking about this next week in detail. They basically go through the bloodstream and monitor, see if there are any foreign cells. If there are any foreign cells, it basically swallows them up and digests them. But if these bacteria have capsules, they're basically indigestible. So the phagocytes swallow them up, try to digest them, can't, and basically spit them back out. Okay. So these capsules are one way that they can physically protect themselves from the immune system. There's also can be components of the cell wall, which basically do the same thing. Certain different proteins that can inhibit the immune system. Bacteria can produce enzymes that can actually increase their ability to infect. So you don't need to know all the details, but like kinases, for instance, digest fibrin clots. So when you have a blood clot, it's made of a protein called fibrin. And so these bacteria can actually produce enzymes that break down that clot so they can get into different areas of the body. Another way they evade the immune system is by constantly mutating. And so the immune system, so let's say you get infected with clone A of this bacteria. The immune system recognizes that there's a foreign substance in your body and sends out the signal to your body to try to eradicate this bacteria. But in the meantime, this bacteria can undergo what's called antigenic variation, where it makes some slight modifications to some of its surface proteins. So while the immune system is now out looking for clone A type bacteria, it's now mutated to clone B. And so now the immune system notices there's a different type of bacteria. It sends out the signal to eradicate the body from clone B. It sends out all its helper cells to do that. And in the meantime, it's mutated to clone C, so forth and so on. So it's constantly making these small alterations to its surface proteins to appear as if it's a different bacteria to the immune system. Okay, so some bacteria actually goes into our cells. Doesn't just stay on the outside, but actually goes into the cells. And they have special proteins and enzymes that they can actually make these little pores in the host cell and actually go inside. So once they're inside the cells, how do they actually cause damage? Okay, what actually is the infection? What's making you feel bad? What's making you sick? What are they actually doing? Okay, so one of the things they do is they actually use the host nutrients. So one example is you have some bacteria that actually use the iron. So the cells need iron to divide and survive, and the bacteria get inside the cells and use up all the iron. So it basically kills the host cells by doing that. There's direct damage. So the bacteria can just disrupt the host cell function just by physically being in the cell. It can live in the cell and produce waste products, either inside the cell or outside the cell, that can damage cells in the surrounding environment and cause them to die. Or they can produce toxins. So these are the three main ways that you get direct damage from a bacterial infection. So we'll talk about toxins a little bit more. So a toxin is a substance that contributes to pathogenicity. So some bacteria itself is toxic. Some bacteria produces a substance called a toxin, and the bacteria itself isn't harmful, but the toxin it produces is. Toxogenicity is the ability of a bacteria to produce a toxin. Toxemia is the presence of toxin in the host blood. So if you suffer from toxemia, it means you have some bacterial toxin in your blood. A toxoid is an inactivated toxin that's used in a vaccine. So in order to prevent an infection from a toxin, you can get vaccinated with a toxoid, which is basically an inactivated version of the toxin. And then you have an immune response against that toxin. And an antitoxin is what you would produce, what your immune system would produce when you had the presence of a toxin. It would form antibodies against that toxin. So there's two different types of toxins, and we'll talk about each one. There are exotoxins and endotoxins. Okay, and they're very different in how harmful they are and how prevalent they are. So you can see from this picture what the main difference is. An exotoxin is a protein or chemical that's produced inside the bacteria that the bacteria then exudes out into the environment, and the effects would be in the neighboring cells. An endotoxin is a toxin that's actually part of the cell wall of the bacteria. So it's not a protein that's made on the inside and then spit out. It's actually part of the cell wall. Okay, so an exotoxin, again, made inside the cell and then secreted outside, mostly from gram-positive cells. It's proteins. Usually does not cause a fever. Usually causes a more local reaction. It can be neutralized by antitoxin. Again, antitoxin is an antibody against the toxin. LD50 is small. So what does that mean, LD50 small?
That means that not very many people die after the infection.
It actually means it takes very little of it to cause death. Okay, the lethal dose of 50% of the people to die is a small concentration. So it takes very little of it to actually be serious. Okay, on the other hand, there's the endotoxins, usually gram-negative. Okay. Again, it's not a protein that's produced inside the cell and then secreted out, but it's actually part of the outer membrane of the cell. It's usually lipid-based, so it's fat-based, not protein-based. Usually these do cause fevers. It does not get neutralized by an antitoxin, and I'll show you why in a minute. And the LD50 is relatively large, which means it takes quite a bit of it to cause death. Okay, so we'll start talking a little bit more about exotoxins. So the main type of exotoxin is an AB exotoxin. Okay. So an AB exotoxin is basically two proteins that come together to form a toxin, protein A and protein B. And so the bacteria basically has a gene to make protein A and protein B. It turns it into mRNA. It gets converted into protein A and protein B, which come together to form a single complex. Protein A and protein B come together to form a single toxin complex. It gets secreted out into the environment, at which point the protein B part of the toxin binds to a receptor on the host cell. The host cell basically thinks it's something else, thinks it's a nutrient, and takes it inside the cell. Once it's inside the cell, A and B actually split up. And A is the actual toxic part of the protein. And the way it's toxic is it actually inhibits protein synthesis. So it prevents mRNA being converted into protein. Okay, so it's like a little bit of a Trojan horse. Okay, the cell thinks it's a beneficial substance that it's taking in. Once it's in, it splits up. The A and B split up. B goes on its way, and A actually causes the cell to die by preventing protein synthesis. Okay, so again, this is the major family of exotoxins, or AB exotoxins. Another type of exotoxins that are membrane disrupting toxins. So what they do, they're excreted out into the environment, and they basically poke little holes in the plasma membrane of host cells, disrupting the phospholipid bilayer, and this can cause major damage and death of the cells. Superantigens are typically endotoxins, and this is why they cause fever, because they usually cause an intense immune response that leads to a fever. So here's an example of an endotoxin. So again, an endotoxin is actually part of the cell wall. So let's say this bacteria gets taken in by a macrophage. This is one of those cells, a phagocyte that basically goes around and swallows up bacteria in order to rid it from the body. In this case, it swallows the bacteria and actually kills the bacteria, but in the process of killing the bacteria, the components of the cell wall get spit back out into the environment, which causes different chemicals to be produced by the immune system, leading to a fever. Okay, so this is why endotoxins can't be neutralized by antibodies, because they only get exposed once they're inside a cell. The exotoxins are actually excluded out into the environment, whereas the endotoxins are inside the cells. So they're not available for antibodies to neutralize them. Okay, I'm just going to go through this quickly because we're talking about a few of the other types of microorganisms. Again, we focus mostly on bacteria, but there's other families that we are considered microorganisms. So fungal infections, typically the two ways a fungal infection will cause harm to the host is by waste products that the fungus produces or by an allergic response from a chronic infection. Protozoas, we'll skip over protozoas. Helminths, so again, tapeworms, roundworms, those types of things. The presence of the parasite itself can interfere with the host function because they usually use the host tissue and absorb nutrients from the host tissue. And actually, the metabolic waste of the helminth can also be detrimental to the host. So just to wrap up, so we've talked about how the bacteria gets into the system, how it causes harm, and now we'll talk about how it gets out of the system, because that's basically how infection gets spread, is by getting back out of the host and going to infect another host. So these are the main 5. The respiratory tract, that's the main one. Coughing and sneezing is a way that the bacteria can get back out and cause further infection. Gastrointestinal tract, feces and saliva. The germ-terry tract, skin, and blood. And if you think of blood, most people think of like a transfusion or something, but actually, something like a mosquito would actually be a bloodborne infection also. So this is a good slide for you guys as a wrap-up and a summary of this lecture. So basically it goes through all the different major portals of entry, how it penetrates into the system, how it evades the host defense, how it causes damage to the host cells, and then the different portals of exit that allow them to get back into the environment. Okay, nice and short. Any questions?
When you talk about adherence, does that apply to all